bachelor's in science in materials, science and engineering from MIT, followed by a master's of engineering in biomedical engineering from MIT, and then went to UC San Diego for a PhD in biomedical sciences. She has multiple publications and has been doing very cool biochemistry in muscle physiology, and she will tell us all about that today. So welcome.
Thank you. Thank you, Dr. Ortiz. Well, so I've come here from across campus from the Department of Kinesiology. Andy Galpin, Dr. Galpin here is the faculty I work with, and we brought a bunch of the kinesiology students along with us. And we're here to talk to the biological science department today, as the nature of the research we do is at this very unique interface between exercise physiology and biochemistry. So one of the main interests in our lab is studying human performance. So and we do this at a molecular and cellular level. And sort of the question is, what are some of these proteins and gene signals that are transpiring to lead to the adaptations that are going on at elite levels of performance, elite athletes? And this is a really interesting question, kind of in a biological science sense, that most of the time we apply our study of biology to the study of disease when we're looking at health, which of course is very necessary. But a lot of times we ignore the physiology of people at the opposite end of the health spectrum. So people who are doing really, really well in something and are pushing the boundaries of human physiology, human performance. And you know, we don't really have a full concept of what health is if we ignore that end of the spectrum. And there could be a lot of interesting things we learn from studying these people that could indeed help other people with disease later down the line. We're really just motivated by curiosity at this point. Okay, well, what is it that allows us to study real live human athletes? Well, the fact is that we are focused on skeletal muscle tissue. And this is one of the few tissues from humans that we can actually biopsy and have people come into the lab, willing, consenting participants, and we can extract some of their tissue. We primarily focus on this muscle here. It's on the outside of your quadricep, called the vastus lateralis. Probably 80 to 90% of muscle biopsy research is focused on this particular muscle. It's very large, it's very well exposed, easy to reach, not a whole lot of arteries going through it, stuff you could mess up. And it is also very reflective of the type of training you do and your exercise habits, lifestyle in general. So we use this specialized biopsy needle. Andy has been trained to do, he does all of the biopsies we do in our lab. It is kind of a legit surgical technique, but it is quite quick, only about a second or so. And we're able to take out muscle tissue, 100 milligrams at most. It's quite small, about the size of a pencil head eraser. But now we can section off the tissue into these little bundles. Some people say they look like tuna steak tartares. And then we kind of pickle those bundles for a week or so. And then we're able to pick through them and isolate out single muscle fibers. So this is another really unique aspect from studying muscle, is that the fibers are, in a sense, single cells themselves. Probably the only cell in your body that you can actually see with your naked eye. That's pretty incredible as well. Another unique property about them is that they have tons of myonuclei. So we're taught most cells have only one nucleus, the control center. But muscle cells are very unique in this aspect, is that they are dotted with myonuclei all across the spectrum of the fiber. And the fiber, I mean, these are only segments that we've cut out of here, but a fiber could theoretically stretch the entire length of your thigh. So we can image the fibers, which we've done in the past, But we can also dissolve them, we can lyse the fibers, and look at the proteins that are inside of them, the proteins that are being expressed, and the proteins that may be activated towards stimuli such as exercise. So this still is an amazing thing to me that we are able to do live in vivo human research. This is kind of a very rare opportunity. All the research I did, standard biochemistry research I did in my PhD is all focused on, either a cell culture or maybe mice or rats, and there's all sorts of really cool, interesting stuff you can do with those systems. But at the end of the day, sometimes you're always asking yourself, does this really relate to humans? Does this really translate to what's going on in my body? And in this case, we can say yes. All right, so another thing we're very focused on in our lab is something called fiber type. So this is defined by a protein called myosin heavy chain, or MHC, which is a very important protein in your muscle that is essentially this molecular motor that is responsible for all of the contractions that you do every time you move any part of your body. And this myosin heavy chain is known to exist in 3 protein versions, or isoforms, that are encoded by, in the human genome. There is the slow twitch, MHC1, the fast twitch isoform 2A, and an ultra fast twitch isoform called MHC2X. So these 3 different protein isoforms, a cell can express any number of combinations of these particular proteins. And so this leads to what we call fiber types that describe the cells themselves, which there are 6 known fiber types found in humans. There are 3 that contain just a pure single isoform, both for 1, 2A, and also 2X. And then there are hybrid fiber types, some that will express 1 and 2A or 2A, 2X, and then another hybrid type that will express all 3 of them. Now, these MHC isoforms are so named based on their actual properties, the speed at which they are performing their motor reactions. And so that actually translates into the properties of the muscle fiber, the cell itself, in that we see increasing contractile power the more fast twitch isoforms are expressed. And we've sort of listed them out here. And another thing about human muscle is that we have a mixture of different fiber types in basically all of our muscles. So it will be this heterogeneous population where we'll have a slow twitch fiber next to a fast twitch fiber, next to a hybrid. And there's some fiber types that will be more prevalent than others. So these 4 here are generally the most common ones we find in young, healthy adults, whereas these two types are a bit more rare, particularly the pure form of MHC2X. So how do we run our fiber typing procedures? Well, probably one of the main experimental outputs of our lab are these very large, high throughput, big gel STS page electrophoresis. So we have the capacity to run 8 big gels at a time, and each of them can be loaded with 26 fibers, so we can type 208 individual muscle fibers to determine their identities, and generally we take about 100 per participant. And we run them overnight, and then like a typical STS page, you get separation of the proteins based on size. So the MHC1 slow twitch isoform is smaller, so it runs down further than the fast twitch isoforms. And we stain for them using a silver stain because there's so much MHC protein in your muscle cells that you can actually see it with a silver stain instead of a Western blot. And then we go through each lane of the gel, which represents a single fiber, and look at the bands and identify the fiber type. This gel is a little bit rare because we found a lot of the triple hybrid here, but essentially to show those different bands resolving. All right, so fiber type, I mentioned that it's a composition in each individual. But the actual composition can vary and is highly dependent on the type of exercise and training that you do. So we typically represent it here as percentages. So somebody could be 45% slow twitch, MHC1, 30% fast twitch, and then a certain proportion of the hybrid fiber types. And are trying to visualize how this works with different types of activities people might do. Our collaborator at San Francisco, Dr. Jimmy Bagley, prepared this figure to sort of conceptualize that. So on the x-axis, he plots the ratio of slow twitch fibers to fast twitch fibers. And slow twitch fibers they're actually a lot more adapted towards endurance types of exercise. So they typically have a lot more mitochondria. They're able to sustain an activity for a long period of time, but a lower intensity type of activity. Whereas the fast twitch fibers are more adapted towards power and speed, strength types of exercises, where you're exerting a lot of force in a short amount of time, high intensity, but it starts to lose its capacity. Typically, these fibers have a lot of glycogen content in them, which depletes pretty quickly. Now here on the y-axis, Jimmy's put hybrids. So percentage of hybrids over pure fibers. Here we have less hybrids versus more hybrids, which actually tend to reflect how sedentary or how active you are. So now he went through a bunch of literature studies where people had looked at different populations of athletes and people and what they do. And we see some things that start to make sense. So long distance runners, endurance athletes, are down at this corner where they have a lot of slow twitch fibers, but they're quite well trained, so they're down here on the hybrids. Whereas competitive sprinters have to do really high intensity work in a short period of time. They have a lot more fast twitch fibers, but are also well trained, so down here on this axis. And then some people who are more sedentary or just recreationally active, maybe they fall in the middle of the spectrum here, but then higher on the hybrids axis. So a lot of these populations have been looked at, but some rather unique sports have not. And one group, Olympic weightlifters, we currently have a study going on in our lab where we took advantage of a very unique opportunity. Last month, there was the World Championships of Olympic weightlifting, as well as the National Championships, held right here in Anaheim. And we were able to recruit, through the efforts of Andy and grad student Nate, who's been working on this project very extensively, a bunch of these Olympic athletes, or lifting athletes, the top ones in the US of their weight division classes, and we're going to look at their fiber type. We think it could be very interesting. It may indeed fall down on this side of the spectrum. Another thing we're studying, which is rather unique, and I'll get to later on in the talk, is we're studying both male and female athletes, which could be another interesting dimension to see if that has an effect from sex. Okay, so to further drive in this point about how fiber type is very dependent on exercise history, perhaps even more than genetics, was another study that came through our lab. A former grad student worked on her thesis with a pair of 52-year-old twins. So after several decades, they differentiated themselves. One had done a lot of marathons, a lot of Ironman triathletes, put in tons and tons of mileage. And the other one, for the most part, was sedentary. So we call them trained twin and untrained twin. And so we took biopsies from them and then looked at, this is in a sense is the fiber homogenates, so a bunch of muscle fibers mixed together, and we're just looking at the proportion of the actual proteins expressed. But even with that, we're seeing a very, very significant difference between these two individuals who have the exact same DNA, the exact same genetics. The trained twin is over 90% slow twitch MHC1, where the untrained twin has more significant proportions of the fast twitch forms, and presumably in a lot of hybrid types. And also, I'll be talking about this protein later, Ampk. We found gene expression to also be very significantly varied between these two individuals. So really driving in the point that fiber type, which was previously thought to be determined by your genetics, is probably a lot more determined by what you do. Okay, and finally, we've got this other very new project that just started. I said we didn't study disease, but we have some new collaborators from Stanford who do, and they look at end-stage renal disease. So patients who, they're pretty old, 65 to 80 years old, and they are on hemodialysis. Some of them had obesity and diabetes that led to the kidney failure. Others are very frail, but they're all having, in addition to the kidney failure, their muscles are starting to atrophy, to waste away. And so they took muscle biopsies and were very interested in what the fiber type of these individuals might be. And so we picked apart some of the samples. The fibers were a little bit harder, frailer to come between, but we got some initial preliminary results that are quite interesting. So we'll go through and type this gel. Here are the 6 fiber types again. We see a number of the slow twitch MHC1s, which we always do, but then, and of course some hybrids as we'd expect, but then this fiber type that I told you is quite rare, the pure MHC2X, we appear to be getting several of those as well, and we can identify it because the band here, I know they're kind of faint, but it's resolving with the top band of the 2A2X hybrid. And then even weirder was this fiber sample. So I said there were these that exist. This looks like a 1-2X hybrid to me, because you got a 1 down here and then a band up at the top. This fiber type is thought not to exist. So we had another biopsy sample that we went through and again saw 2A2Xs and more of the pure 2Xs. We're starting to be convinced they're there. A triple hybrid here and then some fast twitch here, but then I know it's faint, but this looks like another 1-2X. This is pretty crazy. Maybe we do have a 7th fiber type here. And so we could probably think about what's going on here. It seems strange that these patients would have these very weird fiber types, really, really fast twitch. And there are theories about it. It's almost as if, so the muscle is wasting away, and the few fibers you have remaining are trying to do whatever they can to keep some level of function, to be able to get you out of bed. And so maybe that's what they do. They differentiate into this super fast, ultra fast fiber type to be able to do that, or maybe they just have no clue what they're doing, so they're making something really weird like this. All right, so I'm gonna switch gears a little bit now and talk about protein kinases and cell signaling, which we'll all wrap into in a bit. But at any rate, you might have heard of kinases. I've studied them a lot in my PhD work. They are very key parts of cellular signaling processes. And what they are is an enzyme that transfers a phosphate off of ATP and puts it on another protein. And this often works as an on-off switch for that particular protein substrate, which is the target of the kinase. Kinases will also be substrates for other kinases, and this often turns them on so that they can process the signaling event or turns them off in some cases as well. So we're interested in kinases because they're very important to physiological processes. They will take an input, such as a hormone or exercise in our case, and produce various outcomes that all lead to performance-related aspects. All right, so if you had to choose a kinase out there that was the most tied to exercise that there is, it would probably be this one, Ampk. And it is this master regulator of cellular energy homeostasis. So it has been repeatedly shown to be activated by exercise. It's a very hot kinase to study, even has its own conferences devoted entirely to it. But how it works is pretty cool. It is this heterotrimer, what we call. So it is a complex of actually 3 different proteins encoded by 3 different genes. But they all need to come together in order to work. And the subunits, one is a catalytic subunit, so it's responsible for performing the chemical reaction. There's a scaffolding subunit, which links the protein to other proteins or glycogen. And the gamma subunit, which is the regulatory, is kind of like the sensing subunit, decides if the kinase is going to be on or off. And what happens is when you start exercising, you start drastically lowering your ATP in your muscle cells. And so it's getting broken down into these components, AMP and ADP, the single and diphosphate forms. And these bind to AMPK on the regulatory subunit and ultimately change the conformation so it allows it to be more phosphorylated itself and turned on as an active kinase and be able to phosphorylate other proteins. Now, AMPK, not only does it have these 3 different subunits, it also has multiple versions of those subunits, so isoforms. So 2 versions of alpha, 2 versions of beta, and then 3 for gamma. All right, so what are some of these targets, substrates, and functions of AMPK? Well, one of the best known substrates is this protein called acetyl-CoA carboxylase, or ACC. So it is responsible for synthesizing fatty acid chains. Well, phosphorylation by AMPK is actually a negative regulatory site, so it turns off ACC, and now instead of synthesizing fatty acid chains, you can start breaking them down. It has similar effects on another enzyme called glycogen synthase. We turn that off, and now we can start breaking down glycogen and then start using these energy stores to make more ATP rapidly that we need to keep on living. All right, and then there are 2 other substrates that are interesting to study. I'm not even going to read their names because these are examples of horrible biology nomenclature gone very bad. But TBC1D1 and TBC1D4. So these proteins are interesting because they link AMPK into the insulin signaling pathway and ultimately sensitize your muscle cells towards glucose uptake. So taking out glucose out of the bloodstream so we can go make more ATP ultimately. And this is kind of the explanation for this phenomenon called the post-exercise anabolic window, or as many of us like to call it, the window of gains. This is explaining why people like to drink their protein and carbohydrate shakes right after they've been exercising. And it's really because AMPK has been activated specifically in their muscle cells where they've been working out, and has turned on these processes to sensitize those cells to glucose uptake. And now all of those nutrients that they're taking in can go almost entirely segregated to the muscle cells and not to your fat cells during that time. So pretty cool. Other interests in AMPK, I'll just mention, it definitely has clinical interests. Metformin is this very old drug that's been used to treat type 2 diabetes for decades. Now, like a lot of old drugs, people knew that it worked, but they didn't know how it works. And more recently, it was shown that metformin actually activates AMPK. And you think about it, that actually makes sense, because what is one of the best treatments out there for type 2 diabetes is exercise, and what also activates AMPK is exercise. Another drug that was specifically developed to activate AMPK is known as ICAR. And this was an interesting example. About 10 years ago at the Salk Institute, they did a very study that got a lot of press where they treated mice with ICAR, and they found that the mice had a 49% increase in their endurance capabilities from being on this drug. And here's an example of how, when it involves a performance-enhancing drug, that is probably the fastest time that research goes from the lab to directly towards its application. Because I think I heard that it was either the summer or so after this paper came out, they literally found ICAR in the dumpsters outside of the hotels where the Tour de France athletes were staying. No joke. So, of course, ICAR is now on the list of banned substances from many competitions. All right, so coming into our research, kind of now marrying these two things I've talked about here, the fiber types and the AMPK, we're really interested in how does AMPK behave differently in different muscle fiber types. That is really the ultimate question. So I keep saying this term fiber type specific. What exactly does that mean? Why is it important? Well, I'll just walk you through a kind of hypothetical analogy to conceptualize that. So we have this mixture of different fiber types. Well, let's imagine for a second that the slow twitch fibers are marathon runners and the fast twitch fibers are bodybuilders. This is, again, a performance study. And imagine you have a mixed population of these marathoners and bodybuilders that you're studying. And maybe you're measuring something like their time to run 10 kilometers. Well, we know all out the bat that these two different groups of people are going to perform differently on this type of test. And maybe we want to study something, conditions like nutrition effects on their performance. So whether they're running in a fasted state or a fed state. Well, now if we run these studies like the majority of muscle biopsy research is done out there, we wouldn't actually separate them out. We would just have one group that is everybody. And we might ultimately not see any significance between the conditions that we're measuring. Now, what makes intuitive sense to all of us is that we need to measure these groups individually to have them in two different categories, which of course we have a very significant difference in their performance. And then here we might actually see significance from the effects that we're studying. So this point I'm making is that if you go back to the fiber types, you have a very similar situation. We know that slow twitch and fast twitch fiber types have very different metabolic properties. So it makes total sense to separate them out, and especially if we're going to be studying a metabolic signaling protein, to be studying it in the segregated populations. Well, why aren't more labs doing that? What does it come down to? Well, it actually comes down to the methods. So methods for fiber type specific research turn out to be a lot more time intensive, a lot more laborious, and ultimately sometimes not really feasible. So I'll walk you through that. So what you have to do is you separate out the fibers, which we already do to do fiber typing. That's not a big deal. But then if we're going to look at a protein like AMPK, we have to cut them in half. One part goes for the typing. The other half we store separately for the analysis later on. And we have to wait until we get at least 20 plus fibers of each type we want to study, and then dissolve them all together in one pool to make one sample that we're studying AMPK out of. And this is ultimately because AMPK is at such a much lower level of expression compared to myosin heavy chain. And then for the less common fiber types, like these hybrids, that really just makes the study of them very unfeasible. What would be much better if we had single fiber methods. So in this case, we would just isolate the fibers and dissolve them individually and use the same sample for both the fiber typing and then the signaling protein analysis. Well, what's stopping us from doing the single fiber types of techniques? Well, we run into this technique called Western blotting, which of course is the go-to for when you want to analyze proteins out of cells. So I did this last year where I pooled together a bunch of slow twitch and fast twitch 2A fibers in these, 20 plus of each of them, and then also ran a single fiber alongside of them and looked for some of our AMPK isoforms. So we're already seeing some promising results here that the expression is very different in some of the fiber types. Of course, we're getting those ugly, unpredictable, frustrating Western blot effects here. But we're getting no expression, no detection at all out of the single fiber. It's just too low in protein. And so we're up against this problem. How can we lower our limit of AMPK detection? And how can we enhance the reliability and conserve our samples, which are very precious? Well, This technique, Western blotting, I'll tell you, do you know what it has in common with a Walkman? They were both invented in 1979. And we're not walking around with Walkmans anymore, but we're still doing Western blots, you know. And I'm not the only one that's had frustrations with this technique. It is quite widespread, and frustrations are vented through internet memes. But ultimately, it was putting a barrier on the research we wanted to do. We really needed to find a way to get around it because we knew we were going to get really interesting, exciting results. And so what do you do when you're up against a problem like that? Well, you talk to some of your friends. So a good friend of mine who's an entrepreneur in the biotech industry, and she works at this company called Protein Simple. Well, she told me about this very new instrument that they've come up with called capillary nanoimmunoassay, or the commercial term Simple Western. So this sounded like a complete fantasy to me when she first described it, that it's actually a machine that automates an entire Western blot assay for you. And it does that using same principles as a Western blot. So you're essentially separating out your proteins by size, then you're immobilizing them, and then you're probing them with antibodies against your specific target and a peroxide substrate going through. But what it does is that it puts these separation matrices into individual capillaries, which are very long and narrow and are able to concentrate your protein sample so you can get to lower limits of detection. And then it does it automated, so you essentially just pipette this plate that they manufacture with your samples, your primary antibodies, your secondary antibodies, your substrates, your wash buffers. You put the plate in the machine, you snap in a cartridge, you close the door, and you press start. And it runs automatically. And you come back, it takes just under 3 hours, you come back later to fully analyze data. It's incredible. So let's see if this can solve our problem with the muscle fiber detection. So here we have a serial dilution of fibers dissolved, just different fiber concentrations. And now let's look at one of our AMPKs, beta-2, for instance. So beta-2 on a Western blot, we're really only detecting it at the top 3 concentrations of fibers. And then the signal's kind of petering out, getting lost, and ugly Western blot things are happening. Here's what it looks like with the capillary assay. This is incredible. This got us to nearly 20-fold lower in detection. And now we can actually study the single fiber level range, which is just really advances what we want to do. And it's got great linearity, great reproducibility. This was a particularly awesome antibody that had no nonspecific targets otherwise. And the machine ultimately measures everything on like a chromatogram type view, where you're quantifying the area under a curve. Okay, so this was figure 1 from our recent publication just out in the Journal of Applied Physiology. And we were able to, in that paper, show fiber type specific AMPK expression. So I'll just go through a couple of those examples from the paper. Alpha 2 we found to be more expressed in the fast twitch fiber types. Beta 2 was equivalent in the 1 and 2As, but then had some differences between the 2 hybrids here. The most interesting results, though, were for the gamma, the regulatory isoforms, where gamma 2 was highly expressed in the 2A fibers compared to all the rest of them. Interestingly, not so in the 2A2X, which again, a lot of people don't actually separate out 2A and 2A2X, and this really notes how different those fiber types are. And then with gamma 3, this one was very much expressed in the 2A2X, and in fact seems to have a profile of expression that almost reflects the contractile properties of the fiber, kind of correlates directly with that, which is pretty cool. And this has actually been concurs with research from mice and rats, where they found gamma-3 more in fast-twitch muscles of those animals. All right, so we published that. Now we're working on another project, and this one is looking at how AMPK activates in fiber types. And now we bring in phosphorylation and the substrate targets of our enzyme, and we also bring in exercise. So this rather evil contraption, piece of lab equipment over in kinesiology. And we can look at really interesting questions like, what are some of the metabolic effects of high-intensity interval exercise, which seems to work in a different way than continuous exercise. Also, this time period after you exercise, that's when some of the most interesting effects of metabolism are happening, and what could be differences between the fiber types during that period, during the window of gains. All right, so a year ago, I came up with a rather intense experiment. So where we'd take a biopsy at rest, then the subject performs 6 high-intensity intervals on that evil bike at pretty close to all out for 1.5 minutes, then some recovery in between, another biopsy directly after the exercise, a third one 30 minutes post-exercise, and then another one 90 minutes. And so you design something rather sadistic like this. Who, of course, do you choose as your first test subject? Well, this is another unique thing about human muscle research, is that you can be your own lab rat, which is really fun. I had a great time with this. But in any case, here I am in our biopsy lab doing those bike intervals. And ultimately, we're going to look at some of these substrates that are targeted by AMPK, which this one in particular, ACC, has almost a 5-fold phosphorylation in response to exercise. And then, of course, using that beautiful capillary assay technique, getting beautiful figures. All right, so here was some of the preliminary data from my own fibers. So just again, illustrating this point, the mixed fiber model versus single fiber, or fiber type specific. When we look at mixed fibers, we of course see the increase in phosphorylation that we expect. But then in the time points afterwards, there could be a lot more variability going on between the slow and fast twitch fibers that we're not resolving here. So now, when we separate out the fast twitch and the slow twitch fibers, we see some more interesting effects here. So at rest, there's actually already a difference in the basal phosphorylation of ACC. And then with exercise, they both go up and then come back down at 30 minutes to a similar degree. But then the really interesting time point here was at 90 minutes, where in the MHC1 fibers, the phosphorylation had dropped to a level below where it had been basally, whereas in the 2A fibers, it was still maintained, so activity was maintained. So this was really interesting. Of course, if you're going to publish a study like this, you need to do it in multiple subjects. So now we moved on to, we've collected tissue on 6 subjects so far. So these are all high-intensity interval-trained men with these various characteristics. Their fiber type composition is plotted here. There was one guy that was a lot more slow twitch than the other guys, but the main thing that's the same between all of them is that they're very low in percentage of hybrid fiber types, which reflects their training, which is what we were interested in. And then for the time points, I decided, based on my own data, to substitute out the 30-minute time point for a 180-minute time point that I thought would be a little bit more interesting. And ultimately, 5 biopsies is just way too statistic, way too much. 4 biopsies is acceptable. All right, so here's some preliminary data from 3 of these guys, analysis of this substrate, ACC. So right here, we're looking at expression, gene expression of ACC, which we weren't, this is pretty much what we expected. It does not depend on the fiber type and doesn't really change with exercise. Again, this is only 3 ends. We're going to get up to 9 in the end, so these error bars should shrink. But now, looking at the phosphorylation of ACC, this other protein target, we see, again, similar to the effects that I got from my own fibers, where at this 90-minute time point, there's still an elevated signal in the 2As, and then coming back at 180 to a lower level than the 1s. But again, we need some more N's here. The even more interesting substrate target was this one, TBC1D4. And so this one, right off the bat, has a very different expression profile in the slow twitch versus the fast twitch fibers. And then even more interesting is that the expression in the slow twitch fibers actually seems to respond to the exercise, where it goes down and then comes back up after the subject has recovered. Whereas in the fast twitch fibers, it doesn't really change at all. And now we look at phosphorylation of that target, and we have a much higher level of phosphorylation in the fast twitch fibers at basal levels than the slow twitch. And then in the fast twitch fibers, it doesn't really change throughout the exercise, but with the slow twitch fibers, it goes up and then comes back down. And this is a perfect example. Whenever you're doing, studies on phosphorylation of proteins, you always have to divide your phosphoprotein signal over your total protein signal. Otherwise, you could miss effects like this if you're just normalizing it by like a tubulin expression marker. So we're going to need to collect more data here, but we're starting to interpret this as, in this particular substrate, TBC1D4, is much more relevant to the signaling that's going on in the slow twitch fibers and isn't really important in the fast twitch fibers, where presumably another protein might take that role. Quite likely the TBC1D1, the sister protein of this one that's in the same family. All right, so I've talked a lot about how many of these studies are almost always done with mixed fiber samples. Well, another commonality between a lot of these studies you'll see with human muscle is this when you read the methods. You noticed I had also picked a population of men for that study there. Yes. So are we really studying humans if we're just studying men? And this is actually part of a larger problem throughout biomedical sciences, that a lot of labs tend to study more male mice and rats versus female subjects, and has even been picked up by popular media outlets. And it even comes to a more significant level when you look at clinical trials. So for example, Ambien, which is a very popular, well-known drug for treating sleep disorders. Well, when it was initially studied in clinical trials, it was done with a population of men. And when it went to market and people started using it, they found, oh, women need to take a lower dosage. Oh, our bad. So again, these are things you really need to consider. And on our end, we're really interested in female athletes. So we're living in an era where women are going after strength and combat sports and very high intensity levels. They're pursuing muscle, essentially. They want to be strong. It's now, culture is kind of changing. And they're representing these really interesting research subjects as well, and how could their physiology be working in a different way than male physiology, especially since we know that hormones are very, work hand in hand with muscle. Now, there was one study that was done with Ampk and women, where they looked at a population of men versus women during submaximal exercise, and they found some interesting effects here. But again, this was a study with mixed fiber samples, so they didn't really know in the end if this was really a difference between the sexes or if maybe the women had a higher percentage of slow twitch fibers, which could be a very likely explanation for this result here. And also, this was submaximal exercise, What if they were all doing the high intensity, all out type of protocol we're using? So this would be awesome to add a population of women, another group, to the current study that we're running, as we would be immediately able to address questions like this. Well, then the real question comes down to, why aren't we studying women? Well, a lot of people would say off the bat, oh, it must be sexism. Well, it turns out it's not. It's because of this thing called the menstrual cycle that we all learned about in 5th grade health class. So women have a period of roughly 28 days where they experience fluctuations in multiple different hormones and are highly variable, especially two principal female sex hormones, estradiol and progesterone, that reach, intense highs and lows throughout the cycle. And if you look at the methods that paper that did Ampk with women, you read into them, you'll see this. So all the women in that study had a normal menstrual cycle, they weren't on any birth control, and they came in to the lab for the biopsies between day 7 to 10. Well, if you've ever run a study with human subjects before, you know that the scheduling can often get very complicated. The scheduling with the subject, the investigators. Human subjects don't live in the lab. You don't have control over them. And if you're trying to do something here where women don't even know when their period's going to start or end, and you're trying to coordinate that, it's ultimately just a huge nightmare, and nobody wants to do it. And that's really the real reason, is that scientists like to go after the lower hanging fruit. The things that we can study more quickly, more without less controls, that are easier to publish. And of course, men are the lower hanging fruit. Goes without saying. But we really want to change this. I mean, we're already with the study with the Olympic weightlifters doing women and men. And in that study, we're just looking at fiber type, which we don't really expect would change with a menstrual cycle. We've see that as an acceptable risk in terms of variability. But something like Ampk, which has actually been shown in the literature to be activated by estrogen, is a metabolic signaling protein. There's definitely got to be, some hormonal influences on it. This is more of a concern in this case. However, like I said, we really, really want that, that population added in there. And I'm actually thinking now that I'm going to go towards more a specific subpopulation on women who are on a specific type of birth control. All right, so starting to tie all of this in, where do we really want to go here? And as I mentioned at the beginning of the talk, we really want to study elite athletes. And it's something that our lab is very uniquely in the position to do based on all of Andy's connections to various coaches, various athletes. And understanding some of these properties, especially these newer athletes these days who are now combining strength, power, speed, endurance, all of these physical capabilities together in one package, which was previously thought in our field not to really be possible. Either you could be really strong or you could have a lot of endurance. But now they're kind of setting a new bar on that. And again, what could be some of the differences between men and women and how they adapt to that training, what different types of training might work better for them in those types of situations. And again, this is a huge question, but we think fiber type has definitely got to play a big role in this answering this question, and also being able to look at how metabolic properties differ between fiber types. So maybe we might even get switching of these properties at these elite levels of performance. So with that, I'll finish. Acknowledgements, of course, Dr. Andy Galpin, who I joined his lab about 1.5 years ago, and it's been an incredible experience. He runs an amazing lab here, some amazing opportunities for students in kinesiology and biological sciences. We love it when people have biochemistry experience, but we can also teach everybody everything they need to know. Kara Lazauskas is the graduate student working on this project, and she is in fact an MMA fighter herself, so brings this rather unique skill from that and has this insane mental toughness to weather all of the things the punches that graduate school throws at you. Nate Serrano, he's our other grad student who's in charge of that whole Olympic weightlifter study. And then Jose and Ryan, who were graduate students last year, who helped me with this project. We have a lot of other undergrads who help us out. And then Dr. Lee Brown, former director of the Center for Sport Performance, our collaborator in San Francisco, and Tim Geiger from Protein Simple. And with that, I'll move on to questions. I also have my contact info here. Again, Andy and I are always interested in giving people research lab experiences. And with that, we'll take questions. Well, that would certainly be very interesting, but the ethical limitations of that, you have to be 18 in order to give informed consent. So, yes. What? For the biopsy. For the biopsy. Oh, yeah. There's other things we could do with adolescents, but not the types of things I'm talking about here. Yes?
Actually, that brought up a question for me. Would you also suspect that perhaps, since they're still developing and they're still forming these types of muscles, that it might not even necessarily give you accurate results too?
Yeah, that could certainly be a case there. I mean, your fiber type, there have been some studies that show it can change within a process of 2 weeks even. It's very adaptable, and it's again, presumably more when you're younger. It's got to be changing a lot more rapidly.
Did you see any major differences between, or did you ask about different diet types between the same training methods for using them?
Yeah, with the men we're running in our current study, we have been taking some very tight controls on them. No drinking. Usually the hardest one is that they have to abstain from exercise for like 48 hours before the study. Some of these guys are like, oh my God, how can I not train? But yes, we keep a log of everything they've eaten in the past week. Generally, they're all, it's more just we don't want any extreme types of diets where they're not eating any carbohydrates or any fats or something like that.
The hormones affect the women's expression so much, and what do you think would happen after menopause if women are still training?
So that's a great question you've raised. Actually, most of the studies that are done with women out there for muscle biopsies are very often with postmenopausal women, because it gets rid of the whole hormonal variability problem. And of course, there are a lot of labs that are focused on aging of muscle, because that's a huge problem. And so there's often a lot more funding to go after studies with aging populations. But essentially at that point, it's interpreted that, okay, you flatlined on your hormones, it's not going to change. Maybe you're more like how the men are, but of course the men, like testosterone, also varies a lot, but we kind of just ignore that. So.
I had a couple questions. One of them was kind of about the sort of going off of the whole adolescent thing. I was wondering if you had done anything, or like how people's history in terms of like how much they exercise like as a child or as an adolescent before adulthood, can affect this, and like does that affect what muscle fibers are like there in the first place? How much of it is like exercise as a child versus like.
Yeah, that's something I don't think that really has been looked at, but again, it's usually we evaluate people based on the past few years of their training and kind of what they've settled into, those types of habits. But no, I mean, our subjects could have wildly different childhoods. We haven't exactly asked about that. We ask about like major diseases or accidents or hospitalizations and things like that. But I mean, the main idea is that we're all a very malleable system. Like muscle is one of these few tissues in your body that you have some amount of free will over. Like if you want to make more muscle fibers or you want to make them work better, you have kind of an idea how to do that. Other, you know, if I'd like to grow more brain cells, I'm not exactly sure how to go about that. But yeah, again, you can change, you can make up for poor decisions in your past, for sure.
So, what about differences between muscles?
Mm-hmm. Yeah, that's a great question. There are other muscles that are biopsied, especially the soleus, which is known to have more slow twitch fibers. Again, it is still a mixture. It's very different in humans than it is with mice and rats, other species, where in mice, the soleus is almost entirely all slow twitch fibers. So you can just take tissue out of that and basically say, this is slow twitch fiber, and then some of the other muscles are all fast twitch fiber. But yeah, humans, soleus is definitely a higher percentage of slow twitch. We also do gastrocnemius. I'm still learning these terms. I'm not an exercise physiologist. And that one I think has a little bit more fast twitch. Again, they're both in the calf. There's other muscles studied, and again, it's always a composition with humans.
I guess I'm curious, does the type of training that people do, right, if you look across the two muscles, does anybody know what happens?
I mean, across...
So if you were taking, I don't know, somebody was doing something that was primarily upper body stuff,
Mm-hmm.
and you looked at the muscles that you were looking at, does the training have an effect sort of throughout the muscles of the body, or is it only when they're training specific muscles?
Yeah, no, it's more oriented around specific muscles that they're utilizing in whatever type of activity they're doing. And of course, with the Olympic weightlifters, that's awesome for us because that is like one of their main workhorses for that type of activity.
So you hinted upon the fact that the hybrid muscles are more common among people who are not athletes, and then you touched upon it again with the seventh fiber type.
Potentially, we have to confirm that.
So can you expand on what this hybrid really is and how it connects with the sedentary group?
Sure. So hybrid fibers, I will point out to start with, they have been a lot more ignored in our field than the pure forms. Sometimes they're a little bit harder to distinguish, but the method we run with the SDS page shells, not using any antibodies or anything, or immunohistochemistry, that is like the surest way to pinpoint the hybrids. But the hybrids, again, there's different theories behind this. One of them may ultimately be that when you're not giving muscle a training stimulus, a kind of incentive to differentiate one way or the other towards endurance or towards power and speed, the fibers don't really know what to do, so they're kind of in this nebulous state of one or the other. That's one theory, of course. But again, a lot of these theories or dogmas kind of get reworked as the field advances, and especially we haven't studied much with women before. Does the hybrid sort of sedentary versus trained, does that work with them as well, or could it be very different? And different hybrid types. Sometimes we think the 1-2A may be found more in trained people. We've seen that. Whereas maybe the 2A, 2X is more a marker of being sedentary. So yeah, the hybrids are still a huge open area of investigation.
So since you're pointing to the value of isolating the single fibers, is there any studies or is in your scope to metabolomics of a single cell?
Yes, that would be awesome if we find the right collaborations. There are actually some papers out there that are starting to do proteomics and mass spec out of single fibers, and I'm sure you could get the methods going for looking at RNA, PCR. Again, RNA is a molecule that you can replicate more of, so you can make more of it so you can actually study it. Protein, you're kind of stuck with what you got there. But yeah, that would definitely be a really cool thing to do. And just, you know, RNA in particular, I mean, we've been all focused on protein here, but RNA is very interesting in terms of epigenetics, in terms of the things we're studying, these training populations. What might the RNA like total expression profile look like, you know, in those heat maps, looking at all these different genes, be in like an elite athlete population versus other populations of people.